First, I'll talk about our veterinary vaccine development platform. So our mission is really to democratizing mRNA vaccine. And if you look at the mRNA, there are 5 different parts, starting from the cap to the untranslated regions, CDS, to poly-A. The cap analog is the most expensive component of mRNA synthesis. So the way we can help to democratize mRNA vaccines is actually to offer low-cost cap analog solutions. So if you look at the structure, those are a few novel cap analogs from Synthgene. Cap 4 is an open ring. It's highlighted in a yellow circle. Cap 5 is the one with a double bond. Cap 7 is a 6-member ring instead of your normal pentose sugar. And Cap A is morpholinol structure. How do we come up with these novel cap analogs? We have an AI-assisted cap analog discovery platform. Starting from in silico design, we would have, so AI help us to come up with all different kind of structures, and then we would fit those structure with EIF4E, the initiation factor, to see which one give you tightest binding. So that's molecular docking simulation. And then we would have a list of candidates. And from those candidate pool, we look how easy or difficult it is to synthesize this molecule. If the molecule is difficult to synthesize, we also drop them from the pool because the commercial value will not be high. And then after all those exercises, we actually synthesize those molecules, perform a suite of assays, and then we feed the result back to the design, build, test, learn machine learning cycle. So first of all, because those cap analogs are unnatural structure, we need to evaluate how safe it is. So in this case, we inject mRNA cap with commercial benchmark GAG cap, or in this case is cap 4, UNA cap. We inject 2 different mRNAs. We also inject the mice with just cap analog by itself. The negative control is saline solution, and then we measure the weight of the animal, of the mice. They all track very similar to negative control. And then we look at the blood chemistry and tissue biopsy. We don't see any toxicity. So with our lead candidate from the discovery platform, we identified CAP5 to advance to our vaccine program. So this is a VZV vaccine. VZV is a latent virus. When get reactivated, will give you shingles. So we design different mRNA constructs and test them initially, and later on decide on 2 constructs, GND025-1 and -4, to advance to the next level of testing. So both mRNA constructs encode the same antigen. It's, the mRNA is about 2.2 kb. We also cap the mRNA with 2 different caps. One is the benchmark GAG, the other one is our own Cap5. If you look at the quality attributes of the mRNA, that's a top table. The top 2 rows are 2 different constructs by GAG. The bottom 2 rows are mRNA, 2 different constructs capped by Cap5. If you look at the purity, integrity, or capping efficiency, they're all very similar. And then we encapsulate them in LNA. In LNP, we look at the particle size, PDI, percentage encapsulation. They're also similar. So this is just to remind you the difference between the cap is that the double bond. First, we look at the protein expression in HEC293 T cells. If you look at the bar, so the gel picture is on the left-hand side, and the bar, the data in the bar form on the right-hand side. So in both construct 251 or 254, Cap5 clearly give you more protein expression than GAG. And then we did injection in mice. Those are IM injection at day 0 and day 21. And then we collected either blood or splenocytes at 4 different time points. The data you're looking at is basically the GE-specific IgG by ELISA, so those are humoral immunity. The controls are PBS and Shingrix. Shingrix is a commercial GSK shingles vaccine. And then the next 2 bars are construct 251, cap with GAG, or Cap5. And then next 2 bars are, this a pointer? No, I was trying to see if the laser pointer works. It doesn't work. It's okay. So then the next groups are like 254, again, 2 different caps. So I bring your attention to the last group of data. So first groups are day 7 and then day 14, and then after a booster shot, you have day 28, day 35. Just look at the last set of data. So clearly your 254 construct outperformed 251, and then Cap5 outperform GAG. And then we look at the cellular immunity by checking the number of interferon gamma secreting splenocytes. So compared to, so again, there are 4 different data points. The first 2 pair of bars are control. The second 2 are 251 construct, and then GAG Cap5, and then the second construct GAG Cap5. And then if you look at the days 35, again, you see a similar pattern. So clearly 254 construct outperform 251, and Cap5 outperform GAG. And then we also tried the Cap5 ENE in HPV vaccine. So this is a little smaller size. It's 1.8 kb. And we also have 2 different candidates. The top 2 rows are mRNA-1, mRNA-2, using GAG cap. The bottom 2 are mRNA-1, mRNA-2 using Cap5. And then if you look at the 260-280 ratio, the mRNA integrity and capping efficiency, all 4 constructs very similar. Again, we encapsulate them in LNP. If you look at particle size, PDI, and the encapsulation efficiency, they're also similar. And then we perform the protein expression analysis in 293 T cells. Here you also see with 2 different constructs, the mRNA with Cap5 outperform GAG. And then we test the vaccine in mice. So first we engraft the mice with TC1 tumor. And then 3 days later, we gave the mice vaccine. And then we check for the T cell response and also the tumor volume. So if you look at the bar on the graph on the left, so those are basically T cell response. There are 2 groups of data. One group are the mRNA cap with GAG. Those vaccines are given at 3 different dosage levels, 10 microgram, 20 microgram, 30 microgram. And the next group of data are mRNA cap with Cap5. So you can see the T cell response of Cap5 is a little better than the GAG. It's kind of a lot better at the lower dosage level, like 10 microgram. And then if you look at tumor volume, if you do not give the mice vaccine, the tumor continue to grow. For the vaccinated mice, the tumor stop growing, except the lower dosage level of the vaccine capped with GAG. So that's the yellow square ticked up. And then we look at the survival rate. So if you don't give the mice a vaccine, they kind of died around day 25. For the vaccinated mice, you get 100% survival for most of the vaccine, except the low dosage level mRNA vaccine capped with GAG. So here, again, you see Cap5 outperform GAG, particularly at those low dosage level. So those are just give you an idea. You can see what kind of work we can do to showcase our capability. So we can not only synthesize the mRNA, encapsulate LNP, we can also perform testing not only in cells and also in whole animal. So what is our RNA accelerator platform? So it has about five components. One is mRNA raw materials, because we make those raw materials and they're readily available. We also make them affordable to democratize mRNA vaccine. We can offer mRNA design and optimization service. Followed by that, we can do process development, analytical development. We also have our own ionizable lipid and also a screening plate for you to screen the lipid with. We actually developed about six to seven veterinary vaccine candidates and ready for licensing and collaboration with clients. So basically our goal is to sell GMP raw materials, and we'll help customers to get there faster, particularly those veterinary vaccine companies who did not have the mRNA technology platform in-house. So on the sequence design, we not only do the mRNA sequence design, we also help with antigen design, because to get a better mRNA, sometimes you need to put some thought into design antigen. For example, you may want to introduce some mutation. For example, the COVID vaccine, the spike protein actually has 2 protein substitution to make sure the spike protein stay in a pre-formation conformation. If your antigen is or your surface protein is a polymer, you can think about whether you want to do a polymer or monomer or somewhere in between. We also introduce signaling peptide to the vaccine, such as peptide help protein folding, or help peptide help the secretion of the protein. So after we figure out the antigen, we do the mRNA optimization. For this part, you can have different strategies for capping or tailing. We also have our own collection of 5' UTRs, 3' UTRs, and you can choose from. For CDS, we normally would do codon optimization to get rid of rare codons. And we also would do GC content management, basically you want higher GC mRNA with higher GC show higher protein expression. We also take into consideration of thermodynamics by minimized folding energy and also AUP. And all those, there are other factors we look into, and they don't all point to the same direction, so it's a balancing act. So often we design about a dozen mRNA construct for people to choose from, and then we go from there. So this is our LNP screening service. We collaborate with Bowen Lee. He's in Toronto now. He came out of Dan Anderson's app. So he basically helped us to put together a screening plate. So each, like 96-well plate, will contain a unique ionizable lipid with different ionizable head, linker, or the lipid tail. So what you need to do is when you take the plate, you can just hydrate and then add other 3 components plus your mRNA, and then you got your LNP in a plate, and then you can screen. So down the right-hand side, the top panel shows your example of screening result. We screen the lipid in 293 T cells and A549 cells. You can see about a third of the lipid formulation performed better than MC3. And then we took a few candidates, we screened them in mice, and here you see the 3 candidates perform better than SM102. And the Venn diagram basically show like we perform the green circle, the screen ourselves, our client perform a screen at their own site, and then you see a nice overlap. The bottom panel basically is attempts at screening lipids for tissue targeting, for targeting at the immune cells. So if you look at the little red square, the HeLa cells, you don't see a good performance, but then those lipids did well in raw 264 cells. Those are macrophage cell line. And then we took one, we inject in mice, we see, yes, you see better expression in lymph nodes. So those showcase our process development and analytical development capabilities. So the top left quadrant show what we can do to optimize the process. We can do DOE, we can try different template, we can try different IVT conditions to find the best IVT condition for a particular molecule. And then on the right-hand side are the TFF, different TFF conditions, those are downstream purification process. The bottom panels are analytical testing capabilities. The one on the left basically show how we run the capping assay. So capping assay require design of a cleavage probe, also require high resolution mass spec. So we have both capabilities. We can help client to design capping assay. On the right-hand side are the basically your Z-sizer readout of LNP particle size, PDI, zeta potential. We also have HPLC coupled with CAD to detect lipid identity and lipid concentration. So this is just our platform. So one example, actually, veterinary vaccine that in a relatively advanced phase for us is rabies vaccine. So rabies is fatal, and lots of the cases are caused by dog bite. So our vaccine is not, this one is for dogs, right? So the current state is inactivated virus. So the issue with current vaccine is it shows slow humoral immune response, and then the vaccine does not give any cellular immune response, and the protection is little bit short-lived, and so you need to give multiple immunization. So the data you're looking at is actually a virus challenging study. So here we basically, this case, this is mice. We try 3 different dosage level, high dose at 2 microgram per mice. The middle column is 1 microgram, and the lower dosage level is 0.2 microgram. And we compare to the benchmark, the either inactive virus or there's another mRNA vaccine candidate. So we designed about 29 different constructs. So if you just look at the high dose, there are some vaccine didn't work that well. The survival rate is not as high. However, if you look at just the low dosage level, amazingly, we have like 4 mRNA can give you 100% protection at 0.2 microgram dosage level. So this is, so we look at the virus challenge survival of the animal, and then here we basically look at the neutralizing antibody. So my scientist told me the WHO standard will show protection is 0.5 IU, and then if you look at the the neutralizing antibody titer, we have some mRNA vaccine give you very, very high titer. So they're really effective. And then, so I also want to point out this one, the vaccine is 5 microgram, not 2. And then we actually move the test from mice to dogs. I think we did 5 dogs in this case. We stopped. The injection is 25 microgram of the vaccine. Now here, the interesting to note is if you look at inactivated virus, it's a slow, gradual bar, but you start seeing that over 0.5 IU at week 1. Right? And then the neutralizing antibodies slowly start increasing. However, with a 2 mRNA vaccine, you see a very high neutralizing antibody titer, which means you probably see the protection much earlier than 1 week. Probably day 2, you can already see it. So if we do the test again, we probably start taking the sample earlier than 1 week. So basically, the advantage of mRNA vaccines are it's safe. There's no risk of genome integration because they're not dead virus or live attenuated virus. You don't have a risk of it recombined with actually live virus, giving you another strain. The mRNA vaccine also gave you a cellular immunity in addition to antibody response. In our hand, it has a very short R&D cycle. From DNA sequence to vaccine, it will only take us 2 months. mRNA is very easy to manufacture. So 1 liter of IVT can produce 100,000 doses. You can also package different mRNA in a single LNP, so that makes multivalent vaccine possible. So this is, so next one I'm going to talk about GMP raw materials. So we have a large 20,000 square meter GMP facility with multiple clean rooms, and we have large scale manufacturing and prep capability. All our products are made using animal origin free starting material, also our process. This is very important for GMP. We have strict bioburden control, and we adhere to ICH Q7 for GMP manufacturing. The other important things we offer to people is we actually have a DMF filed with FDA. And for those, if you use our raw material, you file your own IND, we'll give you authorization letter to refer our drug master file. To our knowledge, we have at least 2 clients refer to our drug master file in their filing. We also have a pretty large capacity to synthesize the cap analogs, NTPs, modified NTPs. We're not small company, just started doing this. We've been doing this for 6 years. We have a large library of modified NTPs. Very interesting, we start sending samples to clients for testing, and I got some somewhat surprising feedback. We have about 5 clients told us our NTPs actually give them higher yield, and you're looking like at 2 examples. So I actually talked to my scientist, is this real? And then he said yes, so there's one quality attribute we control that give you this kind of result, but we're not going to tell everybody because that's our trade secret. Again, our NTPs are also animal origin free. Just a few words about Areterna. Aret is a Greek word for excellent. So at Areterna, we strive to make excellent or help clients make excellent RNA. The company was founded 9 months ago to serve Synthgene customers outside China. Synthgene was founded in 2018. We have over 100 patents already, all in the nucleic acid space. We have raised over 400 million RMB. We're raising more. The company has over 400 employees, over 200 customers in US, and now I think we have over about 50 customers in US and Europe. So with that, I'm going to stop, and I'll be happy to answer questions. Thank you.